Hello, everybody. My name is Elena Valsecchi. I'm from the University of Milano Bicocca and Mare Center. I'm going to talk about environmental DNA. So let's start by defining it. The environmental DNA is the DNA which is released in the environment by any living organism. It's found in all environmental matrices, so soil, air, and water, and it originates from skin, hair, fur, scales, mucus, feces, eggs, and sperms. It's found in traces. It has a pretty short lifespan, and it can be either in an intracellular or extracellular forms. So what happens now is that we are able to detect these traces and to analyze them. So how do we analyze environmental DNA? Well, we can actually take two different paths. Either do a barcoding analysis, which implies looking at a single species, therefore using species-specific primers. Alternatively, we could use the meta-barcoding approach. This means looking at more species simultaneously within a single sample. This will imply the use of conserved universal primers. And in this study, we are going to use this second approach, and we are going to look specifically at the marine vertebrate, and particularly with a particular interest in marine mammals. Okay, so universal primer, what are they? Well, universal primers are sort of short string of sequences which are conserved within a certain taxonomic group. So in our case, let's say vertebrates. So for vertebrates, when we use this primer in order to analyze informative regions of the DNA, for the vertebrates, for instance, for the taxonomic purposes, the best loci seems to be the 12S and the 16S ribosomal DNA genes in the mitochondrial genome. Okay, so up to recently, there has been a few primers to be published and been used in different groups of vertebrates. But there are some problems. There have been some problems in terms that most of them were either very small, so the piece of sequence to be amplified was quite small, or the opposite was too large. Now, both these conditions are undesirable when working with environmental DNA. A too small fragment means you don't have enough space, enough room to accumulate variability, and therefore you might be able not to resolve at the species level, while too large fragments are not suitable for working with environmental DNA, which often is very degraded, and therefore you find only small pieces. So bearing in mind this, we have recently developed some universal vertebrate, marine vertebrate specific primers able to pick up from fish up to mammals. And these are, there is one that is found in the 12S gene and the other one in the 16S gene. Now then, are these primers working? Well, they do, they are working because we have already used them in the Mediterranean in a study where we have been collecting data samples from ferries. But I go straight to the point. So the percentage of cetacean reeds we got out of this data was only 0.4%. So is this percentage realistic? Well, we think it's actually an underestimates. And we do think so also because there are many different studies showing that species detection may be biased towards more abundant taxa, so in this case, bony fish, to the detriment of the rarer ones, such as marine mammals, but also elasmobranch, that may remain undetected, although present. So this is what actually happens. So this becomes a problem. So what should we do when we want to use eDNA to work on cetaceans? Well, there is a lack of marine mammal specific metabarcode primers. What we have have been done so far in literature is either using the species-specific assays, so therefore the barcoding approach, or has been using a universal fish-specific primer that sometimes are able to pick up also marine mammals, or as in our case, use universal vertebrate primers, but we have seen that there are some problems also in this case, in terms that the resolution is not at the top. So there we come to the study we are presenting now that not by chance was carried out in the Maldives. Maldives, sorry, that is a biodiversity hotspot, but also as far as cetaceans are concerned. So this becomes the ideal scenario where testing marine mammal specific primers. So the aim of our study was to increase cetacean resolution in metabarcoding studies, identify the most suitable region between the 12S and 16S for cetacean detection, to assess cetacean incidence in Maldivian waters in relation to habitat type, and finally to look at the trophic composition of the vertebrate community inhabiting different habitat types. So here shortly is our sample set was of 39 samples collected over 2 years in 2020 and 2021 from the cruise boat White Waves Maldives. And we actually try to have an overview of the scenario, the Maldivian scenario. So we actually try to collect samples from 3 different marine habitats. So atolls, Kandu, so there are the passages between the oceanic water and the water inside the atoll, and the channels. Okay, the molecular strategies we adopt were actually 2. Well, first of all, the identification of the nested cetacean-specific primers. So how does it work? So this is the situation you might find if you align DNA sequences, and you can see in green are highlighted the sites, the priming sites where the conserved vertebrate primer sits. And as you can see, these 2 green sites are flanking hypervariable regions, which allows us taxonomic identification. Now then, if inside this hypervariable region, we are able to identify an area, a motif, which is conserved only among cetaceans, we will be able to selectively amplify on the cetaceans. And the second things we try to do, it was to run in multiplex, that meaning simultaneously in the same run, the 2 amplicons obtained by the vertebrates and the cetacean specific DNA. So this is our results. It's slides showing what we found. So we actually were able to find signals from 10 different species, and here you can see in the columns we have the 4 primer sets. So the highlighted are the one, they are vertebrate specific, so both for the 12S and the 16S locus. And then we have one marine mammal specific, because it will amplify also pinnipeds, and the cetacean specific primer for the 16S DNA. So what kind of information can we draw out of this? Well, first of all, we can see that both the cetacean specific, marine mammal specific primer perform much better in resolved cetaceans than the vertebrate primer. So we have 5 instead of 3 species for the 12S primer. And we have 8 instead of 4 species for the 16S marker. Also, however, if we see the same piece of slides, adding an extra piece of information, which is in how many samples each single species was detected, well, then we can see again that the cetacean-specific primers perform much better. And so they were able to identify the various species in more samples. So the resolution was, of course, much higher. However, there are also some unexpected results. For instance, we find, and actually not in one, but in 4 samples, we find sequences which were annotated as belonging to the genus Sotalia. So this is very strange because Sotalia is present with 2 species, but that should not be around this area. So here I'm asking to the expert in Maldivian cetaceans what can be the case. What the possible explanation on the molecular point of view is that this sequence refers to a dolphin species which is either hasn't been identified yet or whose reference sequencing has not been deposited. So that would be very interesting to go in depth about this point. The other unexpected results is the huge number of rates and that were ascribable to the Balænoptera physalus, to the fin whales. That is not expected in such a high quantity in this area. And also is the only mysticete species that we were able to detect. So the possible explanation is a contamination from the mock template that we use as a positive control that indeed contain fin whale DNA. And possibly at the same time is also hiding some other mysticetes. So also in this case, we should go better, find a better resolution. Also, we find many cases in which the sequences, the reads that we found were assigned to not at the species and not at the genus level, but at the family levels. So that means that we can try to go deeper into these sequences. Okay. So this is another way of showing our results. Here we see the different species and their occurrence throughout our samples. So what we can notice here is that the most abundant species retrieved by in our molecular surveys actually fully overlap with those most frequently encountered in visual surveys. And also, although our samples, our sampling regime was opportunistic, therefore we were not looking for cetaceans, yet in 11 instances, they has been reported from people on board the presence of a Stenella without specifying the species in the surrounding waters. So if we look at these samples, actually in 72.7% of these, either Stenella longirostris or Stenella cerularbra, therefore the spinner or the striped dolphin were detected. And in all but one, there was at least one odontocetes. Well, then there are, as we have mentioned before, there are many cetacean sequences that remain unassigned. And so here there is room for improvements in the technique, definitely. Okay, so we also look at the species abundance in relation to the habitat type, but we didn't find any significant differences. So that's suggesting that a regular use of all habitats by cetacean, that's for supporting what was found by Letizier et al. That used both visual and acoustic data on the distribution of spinner dolphin, seeing that the spinner dolphin tend to be within the atoll during the day, and during the night it gets out to feed. So we have enough data for the spinner dolphin to see what our data were suggesting, the molecular side, and actually also in our case, we find that the spinner dolphin was found in all habitats. So although this talk is mainly focusing on cetacean, as I said earlier, we actually run a meta-barcoding analysis, meaning that we also look at the fish community. So I'm not going to tell much about this, but I wanted just to show up some results which are related to the trophic level. So the trophic level is the position that any given organism occupies in the food chain. So the traffic level in the aquatic environment may range from 2 to 5.5 for specialized predators. So we wanted to look at the distribution of the trophic levels which were assigned to each of the single species that we identify in each of the samples, and to separate them according to their habitat. So what we are seeing here, that while in the atoll and Candu samples, all the 5 different classes of trophic levels are more or less equally represented. Instead, in the channel samples, we mostly detected pelagic and predatory species, which is not a surprise, but it's an interesting way of validating the power of environmental DNA in picking up what is actually the local community that we are going to sample. So in conclusions, in this study, we flanked the use of universal vertebrate primers to that of 2 nested primers specific for marine mammals, and we tried to test their ability to be multiplexed in a single next generation run. So the new developed marine mammal specific primer proved to be efficient in increasing the detectability of cetaceans. They proved to be compatible for multiplexing. We would say that it's advisable to combine both the 12S and 16S molecular data, as each marker is able sometimes to pick different species. So in order to cover the whole, whatever we can find out, it's better to use a combination of both primers. And also, so this is a way that provides an additional tool for a versatile and cost-effective monitoring of vertebrate biodiversity. So what's next? That for sure we are intend to improve the taxonomic resolution for cetacean and to bring them all up to the species level and reduce the positive control contamination risk.
Elena,
also, sorry? This is last
we're
one.
already past 15 minutes.
Yeah,
this is want to wrap up really quickly.
It is the last. It's the last slide. So just we would like to integrate the molecular and visual data, and we will do this thanks to the collaboration with the Maldivian Marine Research Institute, which is hosting this meeting, and Charles Anderson. And so we were able to get 22 extra samples, which were collected simultaneously to sightings. And so we are looking forward to these results, and thank you for your attention.